bamboo corals, and with every one of them was a shrimp species. There were also at least 3 different types of sea pens that we saw in the sedimented area.
And each one of these organisms seem to be pretty much isolated as opposed to living in a clustered community like we've seen on the rocks.
Discontinued that position move, stand by.
Copy.
But it was
Ship is
amusing
holding
to
station.
see in each of
Copy
these
that.
bamboo corals there was a single shrimp living
Or I
among
presume
the tentacles.
they said discontinued the move, so I assume that they're holding position and not drifting. Keep an eye on it on a high pack.
Yeah.
If you've just tuned in, we are hopefully temporarily off the bottom.
It's our off bottom time.
We're experiencing some issues with the ship, and so we
1535 local,
needed to
1935
remove
UTC.
ourselves from the seabed, and we're hoping to return. The good news is that we have covered a lot of the proposed dive, and we've gotten some good samples of some of the characteristic fauna, and also a good rock sample that definitely represents this particular dive.
Because we had it saved the last time, but I mean, between now and then.
Oh, the hardware parameters like set up?
Yeah.
Okay.
That's what I'd imagine. Because we don't have any physical hardware changes.
Okay. Yeah.
As far as I'm aware.
Yeah, I'm just not familiar with the HyPack setup.
You can't do it from here.
You have to do it from administrator, right?
Yep.
Okay.
And not while it's running.
Yeah. Not while survey's running.
Today is different from some of the other days out on our cruise. We have Ask Me Anything questions that are being submitted. We've had many so far, and so we've been trying to answer some of these while we've been narrating the dive, and also folks on shore are answering some of the questions. So we encourage you, if you're familiar with the Reddit Ask Me Anything questions, go ahead and submit a few more. I think we're keeping them open for a few more hours. One of the questions that I've responded to, I've received similar questions related to the mapping of the seafloor. And one of the questions was that it appeared from Google Earth that the entire seafloor had already been mapped, and who did that, and what kind of technology was used, and why do we still need to map the seafloor if it's already been mapped?
Okay, everything's stabilized for us up here. We stopped in the middle of a position move, and we are ready to reinitiate that position move. If you want to update us with the distance and direction.
Low resolution pictures that are blurry, and each of the pixels in your image is huge, so you really can't see the detail. That's basically the kind of map that we currently have of the entire ocean. And that map, and there's more than one, but one of the main maps used was determined from satellite altimetry. And I won't go into details about that, but basically the sea surface height is measured from satellites. And sea surface varies in height, even though you don't perceive it being on a ship, but the heights vary depending on what lies below. And that all has to do with gravity, and
this is Bridge
I won't get into that. It's a little lengthy explanation, but
or would you like to go
basically
slower?
That's a far distant
I'll definitely
view of the
try to get up to 3. I'll start slow and
sea
see if I can build to 3 over.
So what we are now
well, we'd
doing
like to go 60
in our
meters,
expedition
that's 60
and
meters,
all over the globe, ships are now
speed decimal 3.
becoming outfitted with multi-beam
thank you. I'll set that up and
sonars,
call you.
and that way
Thanks.
we can get a much closer look at the seafloor using
ROV
sound.
nav bridge, sorry, I did not read that back to you. Just to confirm, 60 meters, 020 degrees, decimal 3 speed. Over.
Higher resolution camera shot, you get a much more detailed image. So the better detail, the better you can start to figure out what is there and how it got there. As a geologist, we certainly want to know the origins of all these seafloor features and how they relate to plate tectonics, to currents, to all sorts of activities that are on the seafloor. And as biologists, They, of course, want to know where habitats occur, and we do see strong associations between outcropping, hard bottom rock, substrate, and biological communities. So the better look we have, the more questions we can ask, but also we can begin to answer some of those questions that exist.
If we do that, maybe. There we go.
There was a question asked here that I think I would like some of my colleagues to think about, and we've gotten some great broad questions. So this one is, and it never says who the questions are from, so I can't credit the source, but what is the largest question you would like to see solved about the deep ocean?
It's not a camera thing. They're just kind of talking about it in the background.
So would either of you ladies like to grab that one?
I'll have to think about it for a few minutes.
Okay,
and then
we'll come back to you.
Yeah, let me.
My answer, the reason I brought that one up is my answer is that I would like to see better resolution of the seafloor so we can start to answer some of the plate tectonic and other origins of the seafloor features from a geologic perspective.
Yeah. Or just
and that would help guide our knowledge about what biology is there as well. So that would definitely be a very good thing to master over the next 100 years or so.
Yeah.
I think we have a lot of Dayala, right?
The question that we always get from
30 gallons.
Many different people is, what's the most unusual thing you've seen while you've been out to sea?
Hopefully we only have 30. We ordered 100.
Does anything come to mind to either of you?
Where did 70 gallons go?
Is there some in Quonset?
I know you filter through all the unusual things you've seen. What's the most unusual?
I mean, I think in total I've spent like 6 months of my life at sea, so it's hard for me to go back. But it just if I focus, if I'm allowed to just focus on today, since I can think of it, it's like the brittle star that was swimming earlier.
I mean, 50 gallons.
That was something I think none of us had ever seen before.
Yeah, that was amazing.
That was really cool. And like I said, at the time I'd seen
all fresh oil. What did we use? Where did 25 gallons
crinoids swim before?
Because we would fill stuff up and drain it to flush
not
out
uncommon, but not common.
Two Parker movies here, I think.
And that's really cool to see.
Yeah, they have like a ballet dance. They're so delicate looking when they swim.
And the move has been initiated, but I'm just at decimal 1 for speed right this moment. I'll see if I can build a little bit over.
Like lateral.
Yeah, more of a breaststroke.
Yeah, exactly. So that was
one of the
coolest, weirdest things I've seen while at sea.
I would rather just get it locally because the guys that we deal with back there
so
are terrible.
Maybe a couple of top encounters where we've seen billfishes several times, and
you said mineral.
There was a swordfish that came right at the submersible in the Johnson Sea Link years ago in the West Florida slope off of Florida, and it wound up hitting the sphere with its spear, and the spear broke off, and we watched the spear kind of spiral down into the depths, and the fish, I felt sorry for it. That was very interesting. And then I've seen big manta rays come over the top of the sphere and just kind of leave this big black presence there. It was really amazing.
I was on the cruise after you, Cheryl, and we heard about this swordfish attack, so it's really nice to meet the person who it happened to. Too bad you didn't get the sword.
Yeah.
I think that happened to the Alvin at some point, and the sword, this might be a marine myth, but I think the sword was recovered.
Oh, wow.
I seem to recall that, but correct me if I'm wrong, anyone out there who knows for a fact that that's a myth. And thanks, Tara Luke, you have confirmed that it is a fact that Hui has a picture of the swordfish attacking Alvin and losing its bill.
Bridge, this is ROV Nav.
I was wondering if the bill was actually stuck somewhere on the Alvin and it was recovered. That's the myth. I don't want to pass that one on if it's incorrect. So maybe you can let me know.
Yeah, just as FYI for feedback then, moving on that heading of 020, we're fighting against the current, so it was using a lot of power. I was able to do it, but only very slowly, and I'm holding position right now, over.
Copy, yeah, we're going to see if we can get a dive track that'll go toward the southwest with the current.
Okay, thank you.
Well, actually, this is Nav. Yeah, so if you take a look at HiPAC, you can see that, so you can see up at the top right of the screen, that's where we lost ship propulsion and started drifting, and you can see where we are now. So moving along the track that we were actually moving directly against a 0.7 knot current at the surface, which is coming hard off the beam on the ship, which is hard for the ship to fight, moving in that direction. So we're waiting for the word from the dive super to go back down to the bottom, but when we do so, we'd like to maybe just come down here where we're at and then actually move in the opposite direction toward the southwest with the current at the surface, making it easier on the ship. We just wanted to make sure that that would be fine with you guys. We should be within 10 meters. 10 meters of the depth that we were at. And in fact, we could move upslope to that trail of sponges and just follow it to the southwest instead of the northeast. Sure. Yeah, okay. We'll keep you updated. Thanks. We need to wait for the word from the dive supervisor. We're going to hold in the midwater for now. Yeah, thank you.
It's really interesting. In the chat room, there are several other scientists who have had billfish encounters while in submersibles. And Tara Luke actually has a quote here that in 1967, during dive 202 of Alvin, the sub was attacked by a swordfish on the bottom at about 2,000 feet. The fish became trapped in Alvin's skin and was brought back to the surface.
The whole fish?
Wow, the whole fish.
I thought just the sword was brought back, so it's an extended story from what I thought. It's probably on YouTube, Herb Garrison said.
Yeah, and then that happened with Shirley Pomponi as well, and they said that they had got part of the bill came back with them. And then 2 days later, when she and Don were diving on a sinkhole about 40 miles away, there was another attack. And they saw the fish at the bottom of the sinkhole bleeding profusely. Oh, I no longer eat swordfish. Yeah, it's
wow,
a harsh reality there.
It is. Well, it's interesting to know that they are aggressive, though. They didn't like submersibles, or they don't like submersibles.
It's an aggressive thing, or is it that it's shiny and looks like a bait ball?
Well, I have a feeling they might mistake it for fish. Or either that or they're chasing fish that are attracted to the lights and then run into us.
Yeah.
Well, I think attacking a bait ball is somewhat aggressive, too.
Yes. True.
But right. Not just a spirit to eat it.
Right.
We have another Ask Me Anything question from a younger member of our listening audience, and that is, do you ever see sharks? What kinds of sharks do you see, and do you have any favorites? And Stephanie, what do you say to that?
One of the more common sharks that I see and I enjoy seeing are pretty small sharks, maybe 2 feet to 3 feet long, and just various species of deep-sea cat sharks. So they don't look quite like your typical shark with big teeth kind of sticking out of their mouths. They actually, their mouth is generally, well, it's broad, and they have more, I think some of them have more crushing teeth, and their mouth is kind of tucked underneath because a lot of times they're picking things off of the sediment or the bottom, the seafloor. And like I said, they're quite small. So they can be similar size to some of the fish that we see, but those are my favorites.
It's funny you mentioned cat shark, because one of the shark species I've seen, and it may not be a true shark, is the dogfish, which is the chain dogfish. Is that correct? Yeah, we saw a lot of those. You probably did also on the slope, continental slope from the submersible. They're beautiful.
Yeah, it is kind of funny that there's cat sharks and dogfish.
Yeah, I'm a dog person myself.
Yeah, and the shark species that I've seen most frequently at deeper depths is the 6-gill shark. It's a quite large shark, and yeah, very impressive.
Those ones don't look like they're really built for speed. True?
I wouldn't know their top speed, but whenever I've seen them, they're lumbering along.
Yeah, that's what I thought. I've never seen one like live, if you will. You know.
If you refresh that web page, it'll go to high contrast.
So it looks like we have a high school science camp at a group at
No, the other one.
Florida Atlantic University. I'm assuming that's what FAU is. They are at the Expedition Command Center, and I want to say hello to them and hope you've gotten to see some of the video that we showed earlier, and we hope to show you some more. Thanks for joining us.
Right, I'm 20 off the
Jim
bottom.
Masterson, if you want to send us some questions on the chat room, we try to answer them. Including who is asking the question. We'd love to say hello to whomever gives us the question. Give a shout out to them.
So to pick up sonar.
Yeah, see sonar off to my starboard. I have visual on bottom.
Copy that. Do you want me to rotate to you or back up and turn?
Yeah, I'll back up to you.
Copy.
Not yet.
Back up. Do you want to stop here?
What's your altitude?
You're
about 20.
I have a good altitude.
Something like that.
Delta of about 12, though.
All right, delta's good then.
Copy. Slowing down.
Yeah, I would stay where you are.
I'll stop.
I would actually come up a little bit. Do you see yourself in D2's aft cam?
Excellent, thank you.
Yeah.
So as you can see, we're not too far off the seafloor. We've just been hovering above. We haven't been lifting up back to the shift. We've just been waiting. Waiting for the go-ahead.
All right. Stopped, Delta 20, 21.
This is a good view of all the tracks made by the heart urchins. Those are very large sea urchins, echinoderms, and they burrow through the sediments. and you can see their traces left behind. Sometimes these are preserved in the rock record. I've never encountered them, but I suspect they are. And we refer to them as trace fossils because we don't actually find the organism that made it, but we do find evidence of burrowing from an organism, so it gives us an idea of behavior. You can see a lot of them here. There's a sea star down there.
I do it a line out west.
We spent the first
we're going to head
couple of hours
a little bit first to try to find the sponges.
We spent the first couple of hours exploring this kind of soft substrate, very fossil-rich sediments. There's a current here, but not strong enough to make ripples. So it's fairly flat and planar with almost no slope to it.
Looks like we have a bit of a shadow in the middle of the light.
We have lots of great
compared to the outside.
video that we'll show on some of the highlight videos, I'm sure, of several of these organisms that we are zooming over. So if you weren't with us earlier, you can definitely have a good look at them soon when our wonderful videographic team assembles the clips for the dive.
So you'll bring the port one up. Port upper is pointed back at you. Yeah, there you go.
We've got about 30 people who are still hanging on, waiting for us to return to the continuation of the dive. We appreciate you staying on the chat line. You've been great at communicating with us. We really appreciate many of your comments, your clarifications of what species we're looking at. Assisting this geologist in learning some of the biota on the seafloor. So this is a nice, almost aerial view. You can see where we were before by the marks made from the sub, from the ROV. And you can see the transition very nicely here from the sediment plain to where the rocks are outcropping. And even from this distance, the changing habitat and all those bright white spots are just some of the many sponges, and there are also many corals that inhabit this area. And up on the top of this ridge is where we see some of the larger specimens.
Yeah, let's move this direction and get us up on this.
It's a great horizon view. It looks like an army of sponges. And the big ones are on the top. Presumably that's where the currents are strongest, and they're able to capture some of the debris. And sadly, speaking of debris, we have human-made marine debris.
285.
That's rusting, so that must be
All right, 30
iron
meters, 285.
can rather than aluminum. Either that or it's just been there a long time and it's being coated with this iron oxide.
Coming out.
So we have a question from the students who are at the ECC at Florida Atlantic, and one student says, do collected animals have physiological issues when they are brought from 2,000 meters to the surface?
Speed 0.2.
Well, it would depend on the animal, but generally there are many that will do fine. The pressure isn't as much of a problem for them as the temperature. So if they're in an isolated bin on an ROV or a submersible, when they're brought up, there's a good chance that they'll be fine when they get to the surface. Fishes at this depth generally don't have swim bladders, which would be the thing that would expand and give them a lot of trouble on the way up. If you can keep things in a contained box so that they aren't exposed to the much warmer temperatures at the surface, they have a fairly good chance of surviving for a while anyway. And some of the corals that we've collected have lived for years in aquaria of some of the scientists, and that allows us to do experiments about their physiology and testing what happens to their physiology when they're exposed to contaminants such as oil and gas or the dispersant
ROV
that was used in the Deepwater Horizon incident. So, sorry about that. What that was. Little trouble with the audio.
Hopefully that won't happen again. We don't know how it happens. We don't know if we'll do it again or not. So it looks like we are continuing our dive, and this is great news. So issues have been resolved, and we're back at the crest of this feature that we had identified and explored already for several hours, and we've returned pretty much to the top of the crest where the abundant sponge community exists. And instead of going into the current, we are likely to just follow the current. You can see the particles are flowing from right to left, and we had been going into the current before, so I think we'll just let it take us down the crest here.
Yeah, video, I wasn't able to get rid of that flare with the lights.
That's probably the craft.
Yeah.
It's definitely better than it was. But it always seems to be worse on days when we're over sediment. So thank you.
Good shot.
Alright, video's gonna switch out.
Okay.
Great, thanks.
Alright, you can come on in, Emily, when you're all set. Just do a snap on this white guy in the middle.
So we've been seeing this particular sponge that looks a bit like a hedgehog quite frequently on this dive, and I'm not sure that we were able to get a positive identification on it.
No, you can leave this.
Yeah, we believe it's the genus Polymastia.
Okay.
But at some point since we've seen it last, Shirley did make a request that we get a nice close-up of it.
Great. We will do that here.
And then maybe we can zoom in on both the squat lobster and that green sponge, perhaps.
Yeah, that green color is not something you see very often
definitely not.
in the deep sea.
It's interesting.
All right, come on in, video.
That's max zoom.
Copy that.
If Shirley Pomponi is on the line, would you be able to tell us a little more about this sponge?
Let's come on out.
Scott, maybe you could weigh in here.
Sure, I can do my best. So we're looking at a demo sponge, probably the genus Polymastia, and each of those little finger-like columns you're seeing are probably the outflow channels, kind of like in this case. Sponges are suspension feeders or filter feeders in that they have cells on the inside that have cilia on them. They beat, and so they generate a current that moves through the sponge, and they're basically a series of pipes and bring water in, and particles that are small enough for the cells to uptake, they will capture them and feed on those, and then send out the water that's already been filtered. And here it looks like we're looking at a squat lobster.
Scott, where is the inflow if all of those tubes that we were looking at were the outflow?
The inflow holes are smaller than the outflow holes.
Really? Huh.
So you can't see them in the video.
Okay.
And they would be far more numerous and covering the sponge. And there's actually some very interesting kind of engineering dynamics, if you will, about a sponge. And that is, if you look at the combined area of the inflow versus the outflow versus where they actually pick up the food, it essentially creates different rates of water movement, such that the slowest water movement is when the water is moving over the cells that are capturing the food, and the fastest water movement is where it's jetting out of the sponge. And this yellow thing you're looking at is yet another sponge.
So are those the intakes on the side then?
Holes. Any of the larger holes that you see almost universally are going to be outflow holes. Basically, lots of the quotes, pipes that are inside there sort of anastomose together. They join together to form bigger and bigger pipes until the last one that the water flows out of is the largest.
Okay, it's coming out. Let's, we're going to follow this, Fernando, whatever the strike of this feature is, to the west.
Rotate slightly starboard. So we can center up sonar.
So we see some more of those hedgehog-like sponges that we just zoomed in on. That Scott was giving us
maybe
information.
Bring you more on top of me, so split the difference.
Copy.
Yeah, so right now he's moved 23 meters of the 30, so when he's done, I'll just make the next move wherever you are.
Okay.
There's another of those green sponges that we just looked at. Is there any ID on that at all, Scott, or are you familiar with the green one?
There was a name offered before. Hertwigia was the suggestion.
Hertwigia, okay. There's a beautiful specimen there.
There's this may be a new coral coming up, this large white sand. Looks like from the distance, maybe this is in the family Primnoidae.
Let's come on in, video. Go ahead. Okay. Let's come on in. Primnoid. Maybe.
Yeah, very nice. So we get a close-up here. This is yet another octocoral. The octocorals are the group of cnidarians that all the sea fans and the sea pens are in. So this is a type of sea fan in the family Primnoidae. And one of the ways we recognize it, in part, is the color of the branches, but each of the polyps that you see extending from the colony has a series of scales over it. And so if you look at each individual polyp, you can kind of see, identify little lines to them. So they're scale-like, what are called sclerites, calcium carbonate structures. And they almost form like a suit of armor, if you think of a medieval knight over the polyp, and then you can see the tentacles extended at the end. And this is one of the most diverse families in the deep sea. So almost every dive we encounter at least one species from this family. But there are many genera and many, many species, so they're a little bit difficult to identify in situ. One of the things that we do look at that helps us to make an identification is whether the polyps you're looking at, are they curled upwards, or are they curled downwards, or do they come straight out? And that's different genera have different patterns. And these look like they're oriented more upwards, so I would at least rule out the genus Neurella. So this might be something like the genus Calyptrophora or Paracalyptrophora, and we'll see if anybody in the chat room has a guess.
All right.
I might get a specimen view.
Sure. You can see the current whipping by. Gives you a sense of...
And I'm sure you all noticed the ophiuroid that is attached again to this species, or there looks like a couple of them. Yeah.
The lasers on?
Yeah.
Okay, you can kill them.
The distance between the lasers, if you're unfamiliar with our technology here, it's 10 centimeters. We use them as measuring devices.
Let me know when you're good, Emily.
Okay, full wide.
Yep. Coming up. Yeah, so we can just move Sirius along this line. I think Sirius is in a good spot.
So that's a great Brisingid. Is it
Along the line
Brisingid or Brisingid?
Maybe
Brisingid.
Move it over just a little
Sea
bit.
Star, the multi-armed Sea Star.
ROV nav bridge, move complete.
Tomato, tomato.
Thank you. There's another of the green sponges, and Joanne Xavier said she thinks it's a demo sponge.
I'm looking pretty much
Order. Oh, I shouldn't have started this one. Poeciloscleridia, or sclerida.
Yeah, I think that's good.
Poecilosclerida.
Thank you. I'm good at saying limestone, basalt, granite.
Those are easier words, aren't they, than Latin names?
I'm looking maybe like 275.
Copy. 275.
So I'm not sure what this is that we're looking at.
It looks like it's two individuals.
I think it's a stoloniferous octocoral.
Are they individuals? Looks like it's lying down. It's being covered up by sediments.
Looks like little Anthem Estes.
Look at
yeah,
that.
I agree. I wonder if there's a chain of Anthem Estes colonies.
Yeah.
I've never seen that before.
I didn't know they could do that. Scott?
Sorry,
yeah, so that makes me think they have either some form of asexual reproduction or crawl-away larvae. And we have seen something like this on Bear Seamount, which is further to the north.
Oh, yeah.
And it almost looked like colonies were dripping down a rock. You know, you had all these separate ones.
Huh.
It looks like what's going on here.
That's very interesting.
Cool.
It's hard to know if they are choosing to be there or if they're being buried by sediments. Maybe they don't want that.
Yeah, I mean, the way they grow, they typically have a fairly thick, short stalk, and then you can see that rounded red part that has the polyps extending off of it. So each of those would definitely be individual colonies. It's just a question of how they got to that spot. Is it a whole bunch of larvae came in at one time and settled, or are these offspring of the first one that settled?
Yeah, at the far right, you can see the connection between them that is on the edge of the rock right there.
And Andrea Petrini said that it could be Pseudoantha Mastis. And I'm not sure that I know much about those, but Andrea, do you know whether that species is more commonly found in clusters like this, or anything about them, about their biology?
Yeah, while she's answering, I'll say the problem with Anthem Estes and Pseudoantha Mastis is really hard to tell them apart in a video. The characteristics are mostly at the sclerite level, which are things you have to look at under a microscope.
Right. And here, wow, look at this guy.
Beauty.
It's coming out in the head.
Wow, look at this guy.
Is that a brochulid?
Could it be?
The three of us are silent as we don't know what this fish is. If anyone can give us a little hand here. I missed how long he is with the lasers.
We'll
I don't
pull
think
out
they've been on yet.
Oh, okay.
Right, come on
We
out.
don't want to blast him in the
blind the
eyeballs
fish.
now. No.
Let's get lasers.
Should be there.
Oh,
There
so
it is. Oh, he's good long. Pretty good size.
10, 20. Maybe almost a half a meter.
Right, lasers are clear.
Can we get
Maybe
a couple
little
seconds? Just
Okay, it's
right
a
here?
custodial of some type, and Andrea's trying to determine the species if she can. Great, thank you, Andrea. Oopsie.
Okay, video's happy.
Right.
Looks like there's a little chrysogorgid coral that's towards his tail, but then
Would you like to
there's a couple
zoom in on
of
that?
other corals on the right side there.
Like, oh.
Power limits reached on bus A. That one scares me.
Bridge doesn't have mic open.
Come
Thank
on
you.
in, video.
Yeah, we've got another patch of either Anthem Estes or Pseudoantha Mastis. It's a kind of coral, and we think they're asexually reproducing, which is why you get each one of the pink round structures is a separate colony.
We also have a little squat lobster hiding under the rock.
Oh yeah,
there.
And are
and
those
a few little
small
sponges.
Yeah.
Golf ball sponges.
Going back to the cuskeel that we were just looking at, it might be Lucio brachula.
Andrea Petrini sent that in. Thanks, Andrea. You know we need help with fish. Although we all thought it might be.
Oh, no.
We're just too afraid to say.
All right.
There's another one of those cuskills. Let's do a snap in here.
Co-pilot, do you want to?
Oh yeah, second one.
20 or 30 minute move.
A little different
Pretty
color
flat.
to this one, not quite as dark.
This one looks like it might have gotten in a couple bar fights.
Yeah, definitely.
Looks like Zero's drifted a little sideways. Good direction.
Maybe he's going to get in another fight.
Yeah, I think that'll work well.
Bridge, this nav.
ROV nav, this is bridge, over.
Next move's gonna be range 20 meters, bearing 270 degrees, speed 2.0.
Oh yeah, spin.
0.2, sorry.
Roger, I understand 20 meters, 270 degrees at a speed of 0.2 knots.
Right.
Good copy, thank you.
Coming out.
Watch lead, this is nav.
Do this little stock sponge right here. Just to snap.
I just wanted to let you know that we have about 30 minutes left on the dive.
Is that gastropod on it?
There's a gastropod up underneath that. That's beautiful.
Wow, that's a very pretty gastropod shell. And a gorgeous squat lobster as well.
And some great pteropod shells. I mean, littered around. All of those translucent triangular objects on the rock to the left, those are pteropod shells.
Just for everybody's information, we have about a half hour left on the bottom here.
Right. Coming up.
ROV nav.
So if anyone in the chat room is interested in another biological collection, please let us know very soon. If you see something, speak up quickly.
And we've been a similar depth for a long time now, about 1,685 meters. And this seems to be the sweet spot of where we can find hard bottom.
running out of rock too, almost. Just a little thin path of it.
Let's do a partial zoom here as we go by. Yeah, that's good.
See the shadow of the shrimp swimming above.
Just do a snap on the stalk.
Oh, look at that.
Those barnacles?
One is.
Oh, with hermit crab.
And it's a hermit crab, but is the upper,
think the other
those
two
are barnacles. Yeah, you can see the...
See their feeding appendage out there. So these will also be filter feeders. So they're up on this stalk of dead coral, getting off the bottom and getting into the current flow, which will deliver more food to them than being closer to the bottom. Oh, and the bottom of this coral is still kind of hanging in there. Is that a chrysogorgid as well, I think? With the golden color skeleton?
Yep, good call, Cheryl. It's like a very small one, young one.
Okay, come on out.
Maybe we could take another zoom on the urchin. Since we haven't done that in a long time, we may have different viewing audience from who missed the earlier shots. It's a nice
come on in.
Coupling of both one of the sponges we've been looking at in detail and a sea urchin, very common. There's a squat lobster under the rock and some sponges, several different sponge species, including one of those green ones, the demo sponge. These urchins are echinoderms along with the sea stars and the brittle stars that we've seen. We've seen a lot of brittle stars, particularly on the sediment areas.
Come out just a little bit.
All the echinoderms have spiny skin, which is what the word translates to. And they use a water vascular system to pump tube feet for locomotion. So these spines are not the tube feet, but the tube feet are on the base, and their feeding apparatus is on the lower part as well, underneath.
All right, it's coming up.
So he's probably feeding on whatever lives on the shrimp, you think. I mean, not the shrimp, the sponge. Not feeding on the sponge. And we've seen a lot of these sponges that are sort of amphitheater-like, facing into the current. And I suspect that indicates that the current is a persistent current from that direction, and this is likely the western boundary undercurrent that sweeps through this area. We're about 121 nautical miles offshore, so we're well beyond the Gulf Stream, which is a northward flowing current. The western boundary undercurrent is, as the name implies, deeper. And earlier we had someone chime in that the water temperatures and salinities indicated that it might be related to the Labrador current that starts as a surface current and then sinks and contributes to the western boundary undercurrent.
Sirius is still moving.
And these currents exist in water depths of maybe 1 to 5,000 meters.
We're moving towards it, but yeah.
The urchin that we were just looking at, the red pancake urchin, might be Hygrosoma,
yeah, I think the ship
Peter's
just
eye.
Stopped. Might have just stopped.
Copy.
Another 20 when they
beautiful color to them. Surprising to see so much color at this depth where there's no light.
Yeah, let me adjust heading a little bit.
And maybe we could zoom in on that brittle star in the center, center lower portion. We've seen a lot of these on the sediment surfaces, not so much in the rocks. In fact, I don't know if we've seen it at all in the rocks.
Come on in video.
Or maybe if they're in the rocks, they're wrapped around some organism that we haven't seen. Yeah. And see that he's lost the tips to some of his arms, and I know at least sea stars regenerate arms. I don't know if brittle stars do. They're cousins.
I'm going to come out.
I've never seen a four-armed brittle star, so they probably do regenerate arms as well. It's pretty difficult to identify at a species level because a lot of that comes from looking at mouth plates on the underside, as I recall.
More, so I was going to call you in just a sec, but yes, we have arrived.
Copy that. Can I give you a next move so you can input it when we're done with that one?
Absolutely.
Okay, next move is going to be range 20 meters, bearing 260 degrees, speed 0.2.
Okay, I understand. 20 meters, 260 degrees, and decimal 2 speed. Just FYI, with the current, I am only able to get about decimal 1, sometimes decimal 2 in speed.
Copy that.
Thank you.
That's great, pilot. Thanks.
And a comment back to that urchin that we were looking at. We mentioned this earlier, but for anybody who has come into watching the video more recently, there were little white, what looks like booties on some of the spines on the bottom side of the urchin.
2 feet booties.
Yeah, 2 feet booties, and they actually act kind of as snowshoes to help them move across the soft sediment.
ROV nav bridge move initiated.
Thank you, bridge.
Yeah, it would be hard to walk across sediment with very spiny feet.
Yeah, the sediment is very soft. And
is this another bubblegum coral, perhaps?
Yes, it looks like it could be a Paragorgia.
They're easy to spot.
Do a snap on a video.
All sorts of things.
Wow.
In and around, there's a interesting, is that another... Interesting how similar the coloration is. I don't know if that's anything intentional.
Well, it would be good camouflage.
It certainly is.
Just like your house.
Yeah, I wonder if there's some evolutionary trait selecting for that orange color for that particular brittle star.
Oh, and behind we have another Brisingid sea star.
A different species.
Basket star.
Yeah.
Oh, and a picniconid on the back side of the Paragorgia coral.
Right, come on in.
This one's quite a bit smaller than the one we saw previously. which was more red like the Paragorgia too. So this is a different color.
So we have time maybe for a collection. Any suggestions of what we might want to collect?
I imagine we'll need to zoom out and look around a little. We already have one sample of this, of Paragorgia.
Of this, so. Yes. And we have one of the sponges that we've seen so much of. Any other of the... I don't think we need another rock specimen. Can we take a look at the sea star that is over there? It seems different species, maybe, from the ones we saw earlier.
It's definitely very different
color, yeah.
Much paler.
Very elongate arms.
Lots of them.
Beautiful.
Also.
Look at how many arms there are. 3, 4, 5.
Go ahead in.
Count.
ROV nav bridge move complete.
So clear in a second.
Copy bridge. Okay, co-pilot, that move was complete. Do you still want to move 260?
No, let's move.
We've seen something like this. No, it's not. I was thinking it might be a coronaster we saw on the Charleston bump, which is same latitude but a shallower depth, but I don't think that's what this is. Do we have any?
This is another Brasingid. I think this is like the one we saw earlier. The coiler arms up in the water column to feed.
25, so I'll just give you a 25 meter move to be
like the more orange color specimen? Okay, so color is definitely not an indicator.
Bridge.
Well, I don't know that it's the same species.
Oh, I see.
It's going to be
same group. It's a very large group. Multiple families in
230
general.
degrees, speed 0.2.
Roger, I understand. 25 meters, 230 degrees at speed decimal 2.
Good copy.
Beautiful specimen.
Yeah, that's awesome. And several other ophiuroids on the sediment surface. The sea star in the middle of the screen now.
This looks different from the ones we've seen in the IGS.
ROV nav bridge, move initiated.
Copy bridge.
Okay, let's come on out.
We'll have to get Chris Ma to look at the video clips.